Hello, my name is Jonas Béal. I'm from Institut Curie in France, and I will talk about how to emulate clinical trials for precision medicine with causal inference methods. In particular, we will apply that to patient-derived xenograft PDX data. First of all, a few words of context, and in particular a definition. What is precision medicine? It means that each patient is assigned an appropriate treatment according to his or her characteristics. In the case of cancer patients, targeted treatments are assigned according to the molecular profile of patients. In the example below, BRAF-mutated patients will be treated with vemurafenib, for instance, and so on. These strategies can be seen as algorithms, as a function denoted PM there, associating a treatment to each molecular profile. That being said, our question is how to quantify the clinical benefit of these precision medicine strategies. One simple way to visualize what we want to evaluate is to represent it in the form of clinical trials, which we call target trials, the one we would like to carry out. So we have a precision medicine algorithm, then we focus on eligible patients, the one with alteration that we can target, and then we randomize them, as in many clinical trials, either in precision medicine directed arm, in which all patients are treated with the treatment recommended by the precision medicine algorithm, and we can compare this arm with some control arms. We have different possibilities. I just described two of them there. The first possible control is just a reference treatment, so all patients are treated with the same gold standard treatment. Another possible control, called CE3 there, is just random treatment. You compare your precision medicine with the random assignment of the same pool of treatment. But now we don't want to perform those target trials in real life. We want to use data to simulate them. But let me take a simple example to explain the method. If you want to estimate the effect of treatment A on outcome Y, you will have an issue because your estimation is biased by confounder C. So that's why we will use causal inference methods based on counterfactual variables. Let me give you an example. Here, on the left, we have the observed population, with untreated patient in red and treated patient in green. The top hat represents the confounder status. If you compute the average of Y, the average of the outcome, in untreated patient, and you compare that with the average outcome in treated patient, then you are looking at association, because confounder is not especially balanced in both subcohorts, so you cannot say anything about the influence of A alone on Y. That's what we want to do, is to study causation. In this case, what you would like to compare is, on one hand, the whole cohort, if all patients had been left untreated, all red, versus the average outcome in the cohort in which all patients had been treated, all green. In this case, of course, that's the same cohort, so the confounder status is balanced in both cohorts. And this is based on counterfactual variables, but we write y of A, and y of A is the value of y if A is equal to small a, even if that's not the case in observational data. But of course, those ones are maybe difficult to estimate. Before talking about estimation, just a slightly more detailed view about precision medicine, because in this case, we have to use one more variable. We still have our genomic coverage C, or treatment A, but we need to have a variable K, the version of treatment, because in precision medicine, even if you are treated, you can be treated with different drugs. That's how precision medicine works. We need to take that into account to not just aggregate all the effects. So we define a new counterfactual variable, y of A and K, and we define for each patient a precision medicine treatment, KPM, that is a function of its genomic covariate. So one example of statistical estimators we can derive is this one. In this case, it's the expected value, the average, of the counterfactual variable for A is equal to 1, so treated patients, with the version of treatment PM of C, so that means that those patients have been treated with the treatments recommended by the precision medicine algorithm. So this average value is the value if all patients have been put in the precision medicine arm. And you can use similar estimators for the other clinical trial arms to have really the effect of your precision medicine strategies, regardless of the confounder. But how to validate that? One original point of our work is to use patient-derived xenograft. In this case, for each patient, you have one tumor that is divided in different samples. Those samples are implanted in different mice, similar mice treated with different treatments. Like this, you can access the drug response for the same patient for different drugs. That's like accessing the counterfactual variables. So we have a ground truth we can compare our method with. Then an example on PDX. So we focus on some PDX for which we have an example treatment algorithm based on KRAS and PA3K mutations. And in this case, we can compare the estimation we have of the precision medicine effect versus what we can compute with our methods. And we can compare that with the ground truth from PDX. So we have the naive method, that's just association, straightforward association. And then those three methods, TMLE, STD, and IPW, are different implementation of the statistical estimators I presented. In this case, the mean absolute error between ground truth and estimation is lower with our statistical methods. So we have defined those methods, adapted to precision medicine, validated them with the counterfactual values, and maybe we will be able to extend that to human data from clinical trial. This work is available as an archive. We have all the GitHub with the code, and we have also a Earthshiny application to try different simulation scenarios with plenty of parameters.